Hello everyone, so welcome to our first lecture of analytical chemistry, and in this lecture video we will look at the nature of analytical chemistry. We'll try to define what is analytical chemistry, and we'll try to see what is chemical analysis. So at the end of this lecture you should be able to define and discuss the importance of analytical chemistry and its interrelationship with the other fields of sciences and engineering. That is, we're going to look at the relationship or the application of analytical chemistry in other fields of sciences and in other fields of engineering. At the end of this lecture course, we also have to recognize the importance of the scientific method and the steps in quantitative chemical analysis in the field of analytical chemistry. And lastly, we should be able to identify and differentiate the applications or the use of the different methods of analysis. All right, so when we say analytical chemistry, it is a science of obtaining, processing, and communicating information about the composition and the structure of matter. In other words, it is the art and science of determining what matter is and how much of it exists. Okay, so let me just read this very nice description of analytical chemistry as shown in this slide. In here, analytical chemistry is defined as a self-reliant chemical subdiscipline which develops and delivers appropriate methods and tools to gain information on the composition and structure of matter, especially concerning the type, number, energetic states, the geometrical arrangement of atoms and molecules in general or within any given sample volume. As we have previously mentioned, analytical chemistry is the science of chemical measurements. It involves the application of a wide range of techniques and methodologies in order for us to get information on the nature of matter. Now, this information can be qualitative, quantitative, or structural. Alright, so in performing a chemical analysis or a chemical measurement, we usually have a sample of a material. The sample has two parts: the analyte, which is the component that we decide to have an information, and the remainder of the sample is called the matrix. Analytical chemistry has three major areas. The first one is called the qualitative analysis. So in this area, the identification of elements, ions, or compounds present in a sample is given priority. For example, in qualitative analysis, it is performed by adding one or a series of chemical reagents to the analyte. By observing the chemical reactions and their products, one can deduce the identity of the analyte. In other words, qualitative analysis establishes the chemical identity of the analyte in the sample. In contrast, quantitative analysis determines the relative amounts of these analytes in numerical terms. Structural analysis is also a major area in analytical chemistry, and in this area we determine the spatial arrangement of the atoms in a compound, or in other words, we try to study how the atoms are arranged in a given molecule. Alright, now that we know what analytical chemistry is and the major areas of analytical chemistry, let's now discuss the importance of analytical chemistry. Analytical chemistry plays a vital role in the development of science. This figure shows us the relationship between analytical chemistry and other branches of chemistry and the other sciences. Now, the central location of analytical chemistry in the figure signifies its importance and its breadth of its interaction with many other disciplines. For example, chemical measurements are used to assure the safety and quality of food, pharmaceuticals, and water. To assure compliance with environmental and other regulations, chemical measurements are used to support the legal processes, for example, in forensics, to help physicians diagnose diseases, and to provide measurements and documentation that are essential to trade and commerce. Analytical methods can be classified first according to method, and we have two major classifications. The first one of which are the classical methods, which include gravimetric methods and volumetric methods. Now, the other major classification are the instrumental methods, in which we use some equipment. So under this classification, we have spectroscopic methods, electroanalytical methods, and chromatographic methods. Alright, so classical methods are also called absolute methods because these methods are based on the measurement of mass or volume of a substance which is directly related to the actual substance salt or the analyte through a chemical reaction with clear stoichiometric relationship. For example, this substance is called acetic acid and it is a component in vinegar. We can determine the concentration of this substance by measuring the volume of the sodium hydroxide solution, of course the known concentration that reacted with acetic acid. Because we know the reaction between sodium hydroxide and acetic acid as shown in here, it is easy for us to relate the amount of sodium hydroxide to the amount of acetic acid by simple stoichiometry. In other words, classical methods of analysis are considered absolute techniques because there is a direct and simple relationship between the signal, that is the mass in gravimetry or the endpoint volume in titrimetry, and the analyte concentration in the sample. As we have previously mentioned, classical methods include gravimetric methods and volumetric methods. In gravimetric methods, the mass of the analyte or some compound that is chemically related to it is determined. During gravimetric analysis, an excess of the added reagent reacts with the analyte to form a precipitate. Now, the precipitate is filtered, it is dried and weighed. Its mass is used to calculate the concentration or the amount of the analyte in a given sample. Alright, so in volumetric analysis, the solution of known concentration, which is called the standard solution, is used to determine the concentration of an unknown solution. Volumetric analysis are also known as titrimetric analysis or titration. The standard solution, which is also called a titrant, is added gradually or stepwise to the solution containing the analyte from a burette. The key to performing a successful titrimetric analysis is to recognize the endpoint of titration. A change in color of the solution is usually signified as the endpoint of titration. Alright, so instrumental methods are also called relative methods. In relative analytical techniques, the measurement of a sample must be compared to the measurements of additional samples that are prepared with the use of analyte standards. Examples are solutions of known analyte concentrations. In instrumental analysis, some physical property or chemical property of the analyte is measured, such as the ability of the analyte to absorb light. The concentration of the analyte is then related by a mathematical equation, for example, as shown here in this slide. Instrumental methods of analysis can further be classified according to the principles by which the measurements of signal is generated. A few examples of this are shown in this slide. The spectral methods or spectroscopic methods, and sometimes called the optical methods, measure electromagnetic radiation that is absorbed, scattered, or emitted by the analyte. The second major category of instrumental methods is electroanalysis or electroanalytical methods. These methods use electrically conductive probes, which we call electrodes, to make electrical contact with the analyte solution. Now, the electrodes are used in conjunction with an electric or electronic devices to which they are attached to measure certain electrical parameters of the solution. Now, the measured electrical parameters is related to the identity of the analyte or to the quantity of the analyte in the solution. The final category of instrumental methods are the chromatographic methods. This method involves the separation of sample components based on the difference of migration rates. Alright, so let's discuss why until now we are still using classical methods, or why are classical methods not obsolete. Now, the first reason behind this is that the apparatuses required for classical procedures is cheap and they are readily available in all laboratories. Another reason is that in instrumental methods, you normally carry out a calibration operation using materials of known composition such as reference standards, and reference standards are expensive. And lastly, instrumental methods are ideally suited to performance of large number of routine determinations. For occasional non-routine analysis, it is often simpler to use classical methods over instrumental methods. Okay, so another way to classify analytical method is based on the extent of analysis. Now, we can have a complete or exact analysis if we determine the amount of each constituent of the sample. Ultimate analysis is when the amount of each element in a given sample is determined. And proximate analysis is when the amount of certain selected constituents or groups of constituents is determined. Alright, so let's look at these three cases and determine the extent of analysis. Okay, a blood analysis which aims to determine the concentration of glucose, potassium, bilirubin, alkaline phosphatase, etc. To what extent of analysis do you think is exemplified in this case? Alright, so this is an example of a complete analysis because we are determining all the components in the sample. Now, let's look at the second case. We have an analysis of gasoline and the results gave us the percentages of the different elements. Now, to what extent of analysis do you think is in this case? Okay, so this is an example of an ultimate analysis because we are determining the elemental percentage of the sample. Okay, so let's look at the third case. So we are determining the amount of impurity in a pharmaceutical drug, aspirin. To what extent of analysis do you think is exemplified again in this case? So this is an example of a proximate analysis because we are only interested in determining a selected constituent in the sample, which is the impurity. Another way of classifying analytical methods is based on the sample size. Depending on the mass or the volume of the sample, we can have a macro analysis, a semi-micro analysis, a micro analysis, and an ultra-micro analysis. Lastly, we can also classify analytical methods based on the amount of the desired constituent or the analyte. We can say that the analyte is a major constituent if it's greater than 1% of the sample. Now, the analyte can be said as a minor constituent if it is 0.01 to 1% of the sample. We can say that the analyte is a trace constituent if it falls between 0.001 to 0.01% of the sample. And lastly, ultra-trace constituents are less than 0.001% of the sample. Alright, so let's try to apply what we've learned. Let's try to consider this first problem. A 500 milligram sample that was analyzed for its glucose content was found to contain 0.25 milligrams of glucose. Which type of analysis best describes this? Now, we have a 500 milligram sample and that falls under a macro analysis. If we take, for example, 0.25 divided by 500 milligram and multiply it to 100, we will have a 0.05%. So this is a macro analysis of a minor constituent, and that is letter B. Okay, so the last problem for this lecture is that if we have a 5-gram sample and it was found to contain 50 milligrams of the metabolite of marijuana, the analysis involves a blank constituent. So in this case, we have to decide whether the analyte is a trace, major, micro, or minor component. So if we divide 50 milligrams to 5,000 milligrams of the sample and multiply it to 100, we get 1%. And we can consider the metabolite of marijuana to be a minor constituent. Okay, so in this lecture, we were able to define analytical chemistry and discuss the nature and applications of analytical chemistry. And we have also looked into the different classifications of the methods of analysis. In the next video, we will look at the typical steps in a quantitative analysis.